Okay, today I am going to talk about my presentation on an egg forum in Chagask, Ireland for my PhD thesis. Negative energy balance alters hepatic miRNA expression in dairy cows during the early postpartum period. Background. Negative energy balance NEB is a severe metabolic disease affecting high yielding dairy cows in the postpartum period. A previous Athenry study has shown that negative energy balance has pleiotropic effects on liver gene expression in dairy cows. A recently discovered class of non coding RNAs called micro-RNAs can mediate such changes in gene expression. Non coding RNAs represent 98% of expressed genome. Micro-RNAs are classed a class of non coding RNAs with 19 to 25 nucleotides in length. 676 wine miRNAs have been discovered. According to Mir-based version 18, miRNAs act through mRNA degradation and or translation inhibition. They regulate up to 50% of coding gene expression. miRNA regulation of mRNA is many to one relationship where many miRNAs can regulate a single mRNA. Objective to determine the effect of negative energy balance on the expression of miRNAs in early postpartum dairy cattle liver to investigate the potential role of miRNAs in the regulation of hepatic gene differentially expressed as a result of severe negative energy balance, material and methods. An energy balance model was created where the cows were differentiated between mild negative energy balance and severe negative energy balance group depending upon their milking and diet. The cows in the mild negative energy balance group were milked once a day and were given ad-lib silage and 8 kg concentrate, while severe negative energy balance group cows were milked 3 times a day, given 25 kg of silage and 4 kg of concentrate. Six animals were randomly selected from each group and their liver tissues were extracted. So after liver tissue extraction, they were placed in trizol, homogenized, and then pellet paint and chloroform was added. They were centrifuged, and then isopropanol and ethanol were used to precipitate the total RNA out of these tissues. This total RNA was then run on miRNA microarray chip for the microarray expression analysis. For this purpose, Affymetrix GenChip miRNA2 array with Mir-based version 15 coverage was used that has 15,066 probe sets in total out of the 679 probe sets for Bos Taurus. So these microarray chip were then analyzed for gene expression, microRNA expression intensities in the form of cell files that came from these microRNA, which were checked for quality control, and then expression of microRNAs were normalized, and then statistical analysis was carried out to compare the expression of the microRNAs between mild negative energy balance and severe negative energy balance groups. And a list of differentially expressed microRNAs was generated and their targets were predicted. Results. Differentially expressed microRNAs in severe negative energy balance group, a total of 76 differentially expressed microRNAs were found, 51 of which were upregulated and 25 were downregulated. Then a cutoff of 1.5 fold was used to select 10 upregulated and 2 downregulated microRNAs. Differentially expressed microRNAs in severe negative energy balance, out of which the important one were listed here, out of which we further analyze MIR2885 and MIR31. These differentially expressed microRNAs were found to be associated with liver diseases like fatty liver and with liver cell cycle regulation and cell growth. MIR31 has non-binding sites in the 3 UTR of 9 differentially expressed hepatic genes from the same model liver, including those involved in metabolic and glycolysis processes. So, when we ran the target analysis for these mid 31 on microcosm target database of my mRNAs, we found a total of 1171 target genes. And out of these, in our set of differentially expressed genes, These were the ones that were present. So, summary and conclusions. Where negative energy balance affects hepatic microRNAs, direct association between miRNA affected and the expression of their target gene in the liver of where negative energy balance dairy cattle were found. SNPs in microRNA seed regions or 3 UTR regions of target microRNA messenger RNAs could affect response to negative energy balance. Thank you.